This is experimental ALD treatment. I'm Aiden Can.
I'm Hera Tellez-Perez.
And I'm Sophia Perez.
Jared is currently sick and he is not with us at the moment. All right. Introduction to ALD. The research that we did primarily focuses on ALD. It's a genetic disease, and it damages a sheath that's found around nerve cells in the brain and spinal cord. Once that layer is damaged, it can cause impaired body function, paralysis, seizures, and possibly death in some cases. The purpose of our research is to find a way in which we can cure or at least lessen the impact of ALD using at least two methods of gene therapy. And we test these methods and observe to see if they either cure or help people with ALD, or if it hinders them in a way, and if it's not helpful. The significance of this research is that it will serve as a stepping stone for other groups of individuals to put time and research into this and not do it blindly. They will have guidance in where to go and in which methods actually help. So we want to use this research as a way to as an open door so that more genetic diseases can be cured in the future using the techniques that we performed.
So for our research methods, we use CRISPR techniques, both base editing and homology-independent targeted integration, otherly known as HITI. All testing that we did was done on mice to model ALD. Our first technique that we used was base editing. With base editing, it has the ability to target and deliver the code we want and need. We achieved this using ABBS, which were injected intravenously, which is essentially a form of gene therapy in which the viral vectors target and edit very specific genes down to the very nucleotide. We essentially modified this gene by having a Cas9 repair the helix with our own specific set of base pairs. In our second technique, we did use homology-independent targeted integration. The HITI method uses NHEJ, mediated targeted integration, allowing for gene insertion in non-proliferating and proliferating cells. In order to carry out in vivo genome editing, we decided to use AAV9 vectors, one of which was a HITI donor and other as Cas9 enzyme. ABCD1 gene mutated mice were used to imitate the condition of ALD in human patients. We later integrated a HITI donor into cells and used PCR primers. And then right there, you can see the PCI primer. To confirm the integration, a group of mice were injected with the HI donor vector was used as a control group. The mice that were left untouched for a month, after which were euthanized. And then after euthanization, we collected blood and separated a number of organs, testes, kidneys, brain stems, and liver adrenal glands to carry out further analysis.
Okay, so for our first techniques, the results we gathered were pretty conclusive. The first technique was base editing. So as you can see to the right, we used the pedigree to keep track of the inheritance. And so what the results were was that the mice injected with the CRISPR modified AVVs experienced improved health as well as more mRNA production than those that were not injected. And on top of that, one of the male test subjects that was not injected with AVVs suffered from worsening symptoms and eventual death. Moreover, blood and skin samples from healthy and affected individuals were genetically analyzed via northern blotting. The first generation results align with the predicted inheritance of X-linked diseases. So as you can see here on the pedigree, the primary generation indicated that the female progeny of the parental generation was a heterozygous carrier because her male offspring in the third generation received the recessive allele and was thus affected. Individuals treated with CRISPR-Cas9 eventually showed signs of remission and recovery. Okay, here you can see a southern blood analysis of the targeted mouse cells. It uses a probe for exon 1 that flanks the targeted sequence. And then there's 3 targeted cell clones, and they're indicated by the arrows, so ES1, ES2, and ES3.
All right. And the results that we gathered through the HEDI technique pointed towards success. The expression of the ABCD1 HITI mRNA was significantly higher in the group of mice that we treated with multiple vectors than on the donor-only control group. The region staining on the right demonstrated the donor HITI was prevalent in tested areas of the experimental group. The HITI mice showed remarkably lower plasma levels and largely reduced VLCFAs, a stark contrast to the untreated control group. In the image above, or on the right, the results are from a PSR analysis of the integration junctions between ABCD1 and the HITI donor. DNA was extracted from these tissues from the groups of 3 ALD model mice. And to conclude, the results from both techniques were conclusive, essentially. They both garnered good results. Mice who were injected with the modified AVVs, they saw improved health and remission of symptoms, and the HITI injected mice also showed improvement in the form of low plasma and reduced via VLCFAs. I would say the takeaway from the results that we garnered is that CRISPR is helpful in reducing the severity and symptoms of ALD through genetic research. I feel like if more research and time is put into honing and refining the techniques that we performed, then we can improve the quality of life for patients suffering with ALD, but we can also improve the lives of all patients suffering from genetic disorders. This expands the area of research to ALD and similar disorders and hopefully sparks more people to start research in this area. If there was any area to expand and keep researching, I would say probably the first technique was the best because it showed the most promise and it delivered really, really consistent results, and we're really happy with that.